joining us for our April seminar on physical genomics. My name is James Peterson, and I'm the Senior Director of Operations and Outreach here at the Center for Physical Genomics and Engineering at Northwestern University. We're excited to welcome a special guest this month, Professor Harmit Malik. Professor Malik is a Howard Hughes Medical Institute investigator and professor in the Basics Sciences Division at Fred Hutchinson Cancer Center in Seattle, where he leads a research program centered on the causes and consequences of genetic conflict. Professor Malik grew up in India and earned his undergraduate degree in chemical engineering from the Indian Institute of Technology, Bombay. Before moving to the United States for graduate school, he completed his PhD in molecular evolutionary biology at the University of Rochester, where his work on retrotransposons showed that these selfish genetic elements were already present in ancestral lineages rather than being recently acquired, as with viruses, thereby reshaping prevailing ideas about their origins. He joined Fred Hutch for postdoctoral research on centromeres with Steven Henikoff and subsequently established his own lab, focusing on how rapidly evolving genetic elements might drive speciation, genome stability, and disease susceptibility. Professor Malik was appointed an HHMI investigator, elected to the American Academy of Arts and Sciences and the National Academy of Sciences, and awarded the Genetics Society of America's Edward Novitsky Prize for creative, paradigm-shifting work on chromosome biology and evolution. Professor Malik, thank you for taking the time to share your work with us today.
Thank you. Thank you for that very generous introduction.
I want to give one quick note just for our audience. We'll have a Q&A session at the conclusion of Professor Malik's talks. So you can just type questions into the Zoom chat at any time, and we will address them during the Q&A. If you would like to learn more about the research and training programs here at CPGE, please visit us at physicalgenomics.northwestern.edu. Check out our growing video archive on YouTube, or follow us on X or Blue Sky. At this point, Professor Malik, I will turn the Zoom over to you, and thank you again. I'm going to go ahead and
thank you.
Yeah, I'll stop sharing. So you can. All right.
Can you see my shared screen?
Yes.
All right. Well, thank you everyone for joining this Friday morning. Hopefully the weather is as pleasant there as it is in Seattle. I am an evolutionary biologist, so this is going to be a slightly different take on the sort of physical genome. And a lot of the work in my lab is inspired by the fictional character, the Red Queen, who was introduced to us by Lewis Carroll in his books about Alice's Adventures in Wonderland. I'm sure many of you have read these books as a child or reading to your own kids. Alice says to the Red Queen upon encountering her in Wonderland that they've been walking for a very long time and they don't seem to be making any progress. And the Red Queen responds that in Wonderland, it takes all the running you can do to keep in the same place, sort of making the first accurate analogy, I think, to academic science, especially today. We are sort of very interested in the Red Queen for a number of evolutionary and ecological reasons, because this became a very memorable line in the context of how battles emerge between species that are competing for fitness space in the same ecological system. So, for example, predator-prey interactions is a really good example of where the fitness of one species is intimately tied to the fitness of the other. And both these species will constantly adapt in order to sort of keep pace with each other's adaptive strategies, with neither species sort of reaching sort of a equilibrium state, because effectively that will trigger a second wave of adaptation in the other species. This is a very important theorem. The Red Queen hypothesis is the basis for much of the public health work we see around the world today. But labs like mine are very interested in adapting this theorem into more sort of molecular insights. And so one way we've done that is to look at the predator-prey interaction, but more from the lens of interactions between antiviral proteins within the cell and viral proteins. And for example, here we have a cartoon of an antiviral protein that's recognizing some feature of a viral protein in order to exert its antiviral action. This will force a Darwinian adaptation on the virus side to no longer present the epitope required for antiviral protection. And that will basically now mean that the virus is at least temporarily winning this arms race. This forces the antiviral protein to now readapt and basically exert its antiviral action again. So what we have here now are two sort of steps in the arms race between host and viral proteins that are entirely being waged at the level of binding affinity and amino acid changes at the binding interaction surface. And so what drives these sort of arms races is the fact that one party is always on the losing side. And so there's always going to be an evolutionary advantage to be gained by innovation. And so effectively what we are trying to do is look backwards in time in these sequences of both viruses and antiviral proteins to try to reconstruct the steps that took place in these arms races and understand their biochemical consequences. So how do we look at innovation? I'll just do like a very quick sort of aside here to just point out that these arms races are not just occurring in places that you'd expect, like host virus interactions, but they also occur in places that you would not expect. For example, in the interactions between sperm proteins and egg proteins, where you might think that it's actually in the best interest of both sperm proteins and egg proteins to interact with each other so that they can actually facilitate fertilization. But we have the sort of asymmetric nature of fertilization to blame because there are orders of magnitude more sperm produced than egg proteins in most animal species, for example, and plant species. But for egg proteins, polyspermy, where two sperm can simultaneously enter the same egg, is a catastrophic event. So what we have here is again the same battle for binding affinity, where sperm proteins are basically rapidly increasing their binding affinity to maximize their chances of fertilization over their sort of sperm competitors, whereas the egg proteins are moving away from this binding affinity just to give more discrimination and reduce the chances of a catastrophic polyspermy event. So again, it's the same sort of arms race, except this is now occurring between proteins encoded by the same species that leads to the same dynamics and the same red queen-like interactions. The way we quantify innovation in protein coding genes, I'll just do like a quick aside for those of you who don't think about molecular evolution on a daily basis. Here we have three codons of a hypothetical protein, and we can imagine that there are two types of nucleotide changes that can occur in these three codons. We could have changes that do not alter the amino acid being encoded. We call these silent or synonymous changes because they did not change the final product being encoded by this protein coding gene, versus those that will instantly alter the protein product. We call these non-synonymous or replacement changes. Now, it's important to point out that mutation, being a chemical process, cannot discriminate between the outcomes, these two different outcomes. So the difference between the fate of these outcomes is actually decided by natural selection. So, for example, if you're looking at a protein alignment that encodes species that had a common ancestor more than 100 million years ago, your eye is drawn towards those columns in which the amino acids have been conserved despite very high divergence. That's not because mutation has learned to discriminate between these sites, but because natural selection has already purged these presumed deleterious non-synonymous mutations that alter protein function deleteriously. In other words, when we look at reverse genetics, we use high evolutionary conservation as a rough proxy for functional importance, and that's certainly a very successful strategy. But we sometimes make the mistaken assumption that things that are not conserved in evolution must by definition not be important. In fact, my entire lab is interested in case studies in which the apparent rate of amino acid change actually exceeds what we'd expect based on chance alone. This is again happening not because of a mutational bias, but because of a selection bias, where selection has accelerated the rate of fixation of these amino acid changes because they were beneficial in the context of whatever arms race drove their rapid evolution. So in other words, this high conservation is a proxy for function, but this constant, relentless innovation at the amino acid level is also a very important proxy for function. What's nice about having a very simple metric like a DNDS test is we can look at all protein coding genes in the genome, and when we do that, we see an overwhelming signature of purifying selection. For example, if we didn't have any selection for protein coding function, all genes would be on this dotted line of DNDS equals 1. The fact that most genes are leftward of this dotted line indicates that for most genes in our genome, an average amino acid change is likely to be deleterious and being purged by natural selection in this comparison of chimpanzees, rhesus macaque, and human proteins. That sort of fits your intuition because many of these proteins have already been evolving for millennia. So they've already come close to what their optimal function within the cell must be. So it's very unlikely that a random amino acid change is likely to improve function. It's much more likely that a random amino acid change is going to be a deleterious consequence. So what I'd like to draw your attention to is that even in this whole gene average, we have some genes that appear to break effectively the speed limit that is imposed by mutation. These are the genes that I'm very interested in. These are the genes that appear to sort of break the imposition of the mutational speed limit because changes at the amino acid level in these genes were deemed to be beneficial and therefore accelerated by natural selection. As I've already told you, immunity genes involved in arms races with viruses and bacteria are heavily overrepresented here. But today's talk is actually going to focus on other categories of genes that really have no business being in this rapidly evolving class, but actually are in this rapidly evolving class, including genes involved in DNA repair, transposon defense, cell cycle control, apoptosis, reproduction, like what I already showed you with the sperm-egg interactions. But today's talk, I'm just going to focus on genes involved in chromatin organization, given the host organization that invited me here, and especially genes that are critical for mitosis and meiosis. Now, what's really interesting about these genes is that in each of them, we don't actually have an a priori hypothesis about why these genes are rapidly evolving. So the very first question is, why are these genes, despite being so fundamental for the viability of the cell or the organism, really allowed to evolve rapidly? And in fact, encouraged, if you will, to evolve rapidly given this fast evolution signature. What's also really interesting is that the very same gene categories show up not only when we look at primate genomes or mammalian genomes, but also in more charismatic organisms like the Drosophila species that we study in our lab, where exactly the same categories of genes, not always the same genes, but the same categories of genes end up showing the signature of rapid evolution, suggesting there is in fact a pervasive force rather than a lineage-specific pressure that is driving this rapid evolution. This has caused us to foray in a number of different model systems over the last 2 decades because each of these systems provides some sort of unique advantage to study one of these cellular processes within the cell. But today's work, I'm just going to focus on our work in Drosophila. And the sort of way I'd like you to think about this problem is we've all sort of grown up with the view of the cell as the very beautiful sort of Swiss watch-like mechanism where all the gears and parts of the cell really need to perfectly interact with each other in order for the cell to be functional. We know that this view of the cell is actually accurate because we have Mendelian mutations in single amino acids that can actually dramatically perturb not just the functioning of one protein, but actually the functioning of the entire cell, because each of these parts actually interacts with many other parts. But the evolutionary signatures from genomic analysis also telling us that the very same proteins are also evolving in these predator-prey-like interactions where they're also very rapidly evolving. And so these two views of the cell are what we're trying to reconcile with each other. One view would argue that there should be extreme conservation of sequence. The other view leads to very rapid evolution of sequence. And how could we basically have rapid evolution despite the fact that these genes have never stopped being essential and critical for cellular function? So I'm going to hopefully try to convince you about this sort of paradox and how we resolve this paradox with 2 vignettes today. I'm going to contrast 2 genes that are very similar to each other, which are the histone genes, which you're all very, very familiar with because they are the core units of packaging chromatin in most eukaryotes and many archaea as well. For example, we've got core histones, which includes canonical histone H3, which is among the slowest evolving proteins in eukaryotes. The amoeba H3 and the human H3 are nearly identical. They actually only have 3 amino acid differences. In contrast to a histone H3 variant, which is actually exclusively found at the centromeric regions of most eukaryotes, called CenH3, but also called CenPA, where it was first identified in mammals. So I don't have to tell you that chromosome segregation is one of the most critical processes, and high fidelity of this process is absolutely critical for the survival of eukaryotic species. This process is mediated by microtubule attachment to specific regions of chromosomes, these so-called primary constrictions or pinch points, which are called centromeres, where these centromeres organize kinetoproteins that will basically mediate this orderly progression and anaphase separation during cell division. If you were to look at the chromosomes themselves, we would see that most of the chromosomes are actually packaged in canonical histone H3, shown here in green. But at the centromere, we have these unusual centromeric specific nucleosomes in which the CNPA moiety replaces the H3 moiety within the nucleosome. And here's where the paradox really becomes stark, because H3 is essential, but so is CNPA. But whereas H3 is among the slowest evolving protein within the cell, centromeric histone H3 or CNPA molecules are among the fastest evolving proteins in many organisms, including Drosophila, plants, and animals. And so really the core question is, why is there rapid evolution of this absolutely fundamental protein that is critical for chromosome segregation? This sort of forces us to confront all the processes that this protein must carry out. And obviously, for most people, the first process they think about is mitosis, because that is like sort of the most common mechanism of chromosome segregation that we think of. And mitosis, of course, is absolutely critical, and especially in a multicellular organism such as our cells, we have millions of mitoses going on every day, and it's really important that each of these processes is well policed. Otherwise, that could lead to aneuploidy or cell death. But from an evolutionary standpoint, this is a relatively boring process. And I say that with all sort of respect to mitosis, because the end products of mitosis are genetic twins of each other, and there's no genetic discrimination possible at the level of which chromosome is inherited by which cell. Now, there are exceptions to this. For example, we have asymmetric inheritance, etc., in many cases, both based on nuclear sort of episomes as well as on cytoplasmic organelles such as mitochondria, but that's not sort of really the topic for this discussion today. In contrast to mitosis, I want to draw the analogy to the contrast with meiosis, in which we basically have two rounds of division which give rise to haploid products that are not genetically identical, but instead are different combinations of mom's and dad's chromosomes, shown here in red and in purple. So now there's actually ample opportunities for there to be a genetic discrimination that can dictate the fates or outcomes of one of these meiotic products relative to the other. So in animals and plants and fungi, all of these should have an equal opportunity to become gametes. Each of them should have the opportunity to become sperm or pollen or spores. But actually what ends up happening is that there are many, many cases in which genes located on one chromosome, let's say the red chromosome here, will encode a poison that prevents specifically the maturation or purple chromosome containing gametes to the next generation. What that means, though, is that even though this male is heterozygous and should transmit 50-50 ratios of red and purple gamete to the next generation, it's only the red gametes that are making it through, which is a tremendous advantage to all genes linked to the red chromosome, to the particular toxin gene. Obviously, things are very bleak for the purple chromosome because it's bearing the brunt of this sort of killing action. But actually, if you were to think about it, all the other chromosomes in the cell have also suddenly lost 50% of their male fertility because of the selfish action of one chromosome. So this is the quintessential selfish gene paradigm, where one chromosome is benefiting and everybody else in the genome is suffering. And that creates this conflict within the genome between how to sort of engage or suppress this selfish behavior. And this is actually occurring many, many times. In fact, there are Drosophila species in which there are three completely independent such driver suppressor systems that are active. And often the suppressor system takes place of an RNAi gene that basically suppresses the production of the selfish gene, and that basically resets the cycle and starts it over again. So even though we think about Mendelian segregation as sort of a uniform entity in reproduction, it actually turns out that there are many deviations from Mendelian segregation, and there is desperate sort of restoration of Mendelian segregation whenever organisms do deviate because of selfish behavior. But what I would also like to point out here is that this is cheating that is occurring. but this cheating is occurring after meiosis is already complete. You're simply dictating the outcomes of the meiotic products and deciding who's going to win versus lose. So this is very unlikely to be the process that gave rise to the rapid evolution of centromeric DNA and centromeric proteins. In contrast, female meiosis, which is this process that has originated at least 12 different times in eukaryotic evolution, is very much the same process as far as the process of meiosis is concerned. But the outcomes of meiosis are very different because in both mammals and plants, only one of these products is destined to be included into the oocyte and be passed on to the next generation. So now the process is not about basically killing your brother's sperm as it was in male meiosis, but it's about outracing the competition to reach whatever preferred position there was that allows you to be sort of passed on to the next generation at a higher than Mendelian frequency. So many years ago, when I was a postdoc still in Steve Henikoff's lab, we imagined that this would occur with any loci that are able to subvert chromosome orientation or segregation. And we thought that centromeres, being the primary location of microtubule attachments, would be the epicenter of this process. So, for example, if the purple chromosome was able to recruit more microtubules to itself, it would orient to this preferred position and reach this sort of preferred position and be included in the oocyte in the next generation. Now, what's really dramatically different between cheating that occurs during male meiosis and female meiosis is that in female meiosis, there's no cost to female fertility because you're producing exactly the same number of eggs, unlike the sort of drop in sperm number in male meiosis. So all you're doing is you're genetically changing the composition of the eggs to favor the transmission of the selfish centromere in this case. Moreover, I think it's obvious to you that this process is actively taking advantage of the machinery of chromosome segregation in order to cheat. So this could be the process that could be the explanation for why fundamental steps of the chromosome segregation apparatus appear to show the signature of unexpectedly rapid evolution. It's worth also sort of thinking back to the fact that another process that allows this kind of rapid evolution to take place in plants and animals is that in both of these species, the definition of what will become the microtubule attachment region or the centromere is actually not determined genetically. In fact, there is like sort of almost homogeneous arrays of satellite that go between the centromere and the surrounding pericentric silent heterochromatin, but instead is actually dictated by where heterochromatin proteins versus centromeric proteins lie down. So if you were to overexpress heterochromatin proteins, for example, you can weaken the centromeric region by encroaching into centromeric space. And if you overexpress centromeric proteins, you can widen the sort of locus in which the centromere assembles. And if you're not careful, you could even try to establish centromeres in euchromatin, which would, of course, be catastrophic from a dicentric standpoint. So in this sort of dynamic centromere heterochromatin compartment that's actually dictated by mass action, Steve Henikoff and I proposed that any kind of genetic change of the centromeric DNA, either in terms of mutation or in terms of recombinational expansion of DNA satellite, would give this particular chromosome a higher propensity to recruit centromeric proteins to itself, with centromeric proteins being the primary determinant or the currency for cheating, if you will, in female meiosis. Once this happens, this particular chromosomal variant is going to outcompete all the other chromosomal variants in the population and potentially drive to fixation. The only way for the other variants to sort of restore parity is for them to evolve altered DNA binding preferences of either the centromeric or the heterochromatin proteins to sort of reestablish the original centromere heterochromatin boundary. So if you were to look at the first cartoon here and the third cartoon here, you might be tempted to believe that nothing interesting has happened here because you've come back and the centromere heterochromatin boundary is roughly in the same place. But you would have missed an intense episode of adaptation, first at the DNA level, followed by adaptation at the protein level in terms of its DNA interaction interface. We can actually make the analogy between this system and a pathogen that learns to invade a host population by learning to bind a host protein. And the only way for the host to restore its own fitness is to move away from this binding affinity. In this case, the pathogen happens to be centromeric DNA, which is completely essential for chromosome segregation, and the immune equivalent happens to be the centromeric protein, which is also completely essential for chromosome segregation. And not only that, these are actually very important collaborators for the process of chromosome segregation in every process of cell division, except in female meiosis, where the asymmetry of female meiosis and its outcomes has basically taken these collaborators and made them into evolutionary competitors. When we first proposed this model called the centromere-drive model, there was some resistance from both our centromere colleagues as well as our evolutionary colleagues. But luckily for us, beautiful work from Mike Lamson and Ben Black's lab using Robertsonian fusion chromosomes in mice has actually shown that the first step of process is completely correct and revealed the cell biology of this process, where expansions of centromeric DNA satellites indeed can recruit more CNPA molecules to itself, and that confers a transmission advantage in female meiosis. And Takashi Akera has elegantly worked out the mechanisms by which this happens. What remains an open question, though, is what is the deleterious consequence associated with this expanded state that leads to the rapid evolution of the centromeric proteins? Because I've already told you that there's not like a difference in female fertility, so there must be something else that explains why these proteins are also rapidly evolving in spite of being completely essential. So this is the sort of the arms race that interests us the most, which is we think, an arms race for transmission bias between selfish centromeric DNA that are the cis-acting sequences that determine centromeres, and centromeric proteins such as CNPA that are the trans-acting determinants of centromere function. Now, I did tell you that this is a rapidly evolving sort of arms race, but I just wanted to sort of clarify that unlike viral arms races, which can actually occur in the matter of hours or days, this is very much an evolutionary arms race, which means this is not like amino acid changes are occurring on a daily basis. These amino acid changes are taking place on the order of millions of years of evolution, which means that it's actually going to be a very difficult project to wait for the next amino acid change to occur and to understand its consequences. So the approach that two very brave souls in my lab took, Ida de la Cruz, who's our lab manager, and a former technician, Emily Colo, is we decided to reverse the evolution of the existing centromeric histone in Drosophila back to an ancestral state, hoping that that would actually unleash some deleterious consequence, which would tell us what drove the evolution in the first place. So in other words, if you think about this as a two-step dance, where the centromeric DNA evolves and then the centromeric protein evolves, we are now basically taking a step backwards with centromeric histone evolution, hoping that that'll actually tell us what was the driving force behind this evolution in the first place. This is a series of experiments we call reversing SID evolution, because SID is the Drosophila ortholog of human CNPA. So the way we do this is we take advantage of the beautiful sort of advances that have occurred because of CRISPR-Cas9 engineering, where we can actually make an allelic series of nearly isogenic Drosophila melanogaster strains, which are identical except at their SID locus, where instead of the original SID gene, we either have a recoded flag-tagged melanogaster allele or a similans allele, which is the next closest species possible, or an ancestor between these two, which was the presumed ancestor because before these species split apart. I just want to make the small point here that similans and melanogaster last shared a common ancestor 2.5 million years ago, and 21 amino acid changes have already happened since then, just in the last 2.5 million years of this 200 amino acid protein. That's 10% amino acid divergence for an essential histone protein, just to give you an idea of scale of what I mean by rapid evolution. So it's actually worth considering, before I show you the results, what we would expect. We could expect that there could be some deleterious consequences in mitosis. This was my least favorite hypothesis because, like I told you, mitosis is about genetic twins going up against each other. My money was very much on male meiosis, that even though female meiosis has no consequence in female in terms of fertility, perhaps mismatches between centromeric proteins and DNA would lead to some sort of male meiotic defect that would lead to lower male fertility. But actually, a third possibility was suggested by work from Elaine Dunleavy and Christian Lehner, that actually the centromeres are the very sort of rare histone that needs to be preserved on the sperm of animal chromatin, because if you were to erase this epigenetic mark, it is very difficult, in fact, impossible to reestablish de novo centromere identity on paternal chromatin. So, in fact, it is possible that what we were going to screw up is this centromere identity inheritance, either in the male pronucleus or in the female pronucleus, which would also lead to lower fertility. So I'm going to show you this through a series of crosses in Drosophila. Since most of you are not Drosophila geneticists, I'm going to use these cartoons to indicate what I'm talking about. So we have two gametes here. We have the Drosophila sperm, which is completely devoid of cytoplasm, and the only SID protein is coming in on dad's genome at the centromeric regions. We, of course, have the maternal pronucleus, which also has SID protein on mom's centromeres, but actually the vast excess of SID protein is deposited into mom's ooplasm because the initial rounds of cell division take place without any transcription or translation in the zygote. So mom is basically supplying all the goodies for the initial rounds of cell division. So after all our CRISPR engineering, we just needed to make sure that we have not screwed up some regulatory features of SID. So the red dots indicate the original melanogaster-like replacement. And when we did that cross, luckily for us, we recovered progeny in the exact Mendelian proportions, suggesting that our CRISPR engineering was successful and did not cause a deleterious consequence that we didn't intend. Next, we basically crossed two heterozygous parents to each other. Now, in this cross, one allele is the original SID endogenous allele, and the other allele is this replacement allele, either from the ancestral species or from similants. And if you remember your genetics 101, you'll recall that we expect three types of progeny here. We expect the original melanogaster fly, which is capable of encoding melanogaster SID only, or the parental-like fly, which is still heterozygous, so makes both types of SID protein. But we could also expect to see flies which are entirely Drosophila melanogaster but have lost the ability to encode melanogaster SID protein. They only encode either ancestral SID or similant SID. Now, our hypothesis is that these flies would be at some disadvantage if they actually result at all. But in fact, what we found was we did recover these flies, but in the case of the melanogaster replacement allele, we got them in Mendelian ratios, but with the ancestor or similants, they were about at 50% or 2/3 of the expected ratios, which means that even though these flies are completely identical and the only difference in them is the SID allele, there's already a viability disadvantage when we score these as adults. This viability disadvantage can be explained because this is a developmental delay caused by the single gene replacement. So, for example, with the original melanogaster flies, we basically see 11 to 13 day sexual life cycle where you basically go from zygote all the way to a sexually mature adult. This is what any self-respecting melanogaster strain looks like at 25 degrees. But you can see that if we have even one allele of the ancestor or similants, that is delayed significantly. And if we actually have both alleles of the wrong type, that's delayed even more. So that means that we basically have a persistent delay in development that sort of manifests as a result of having the wrong SID allele, which is just different at a few amino acids at critical positions. What about fertility? Remember, this was my favorite hypothesis, that fertility of these replacement SID males or females would be affected. So to do that experiment, we took regular wild-type melanogaster females and crossed them to Drosophila melanogaster males that only encode the wrong SID, if you will, either the ancestor or the similant SID. And if my original hypotheses were correct, we would presume that these crosses would not be successful because the males would be either infertile or subfertile. But actually, they were completely fertile. So that instantly eliminates both my favorite and my next favorite hypothesis, which is male meiosis does not appear to be affected, and centromere identity inheritance also does not appear to be inheritance. Otherwise, we would basically have not recovered a successful progeny from these crosses. So then we turn to plan C, which is what would happen if you were to take melanogaster females that only encode the wrong SID and cross them to melanogaster males that are sort of wild type like. Again, these crosses are completely successful. So, sort of like the mini depressing result here is that, in fact, these replacement SID alleles, even though they have a dramatic viability disadvantage, they do not appear to have a fertility disadvantage in terms of meiosis or gamete inheritance. Someone very wise at lab meeting pointed out that we had not actually done the obvious control, which is to take these fertile males and these fertile females, both with the wrong SID alleles, and cross them to each other. But when we did that, what we found was we recovered almost no adults. This is a very, very penetrant sort of phenotype. And these are the sort of the results from that, where if we take melanogaster replacements, we get hundreds of progeny, just like any self-respecting melanogaster strain. But with the ancestors or with the similans replacements, we get basically no progeny. These few progeny that we get, about a dozen, don't actually go another generation. So they're basically dead. So what this actually tells us is the very first and direct evidence that the ancestral or the ortholog SID from the related species is mismatched in the Drosophila genome and cannot support sort of viability of the embryonic development. And so we can instantly ask what is actually happening. Where are these zygotes dying? And in fact, they're actually dying very, very early in embryonic development. And we can visualize that with these sort of images and videos that I'm going to show you, where each of these represents a syncytial Drosophila embryo, which means that there's no cellularization yet. The entire embryo acts as one cell, but each of these dots represents individual nuclei that are going through synchronous rounds of mitotic divisions, as you can see here, but they remain at the periphery of this sort of embryo, and they just sort of populate the periphery of this embryo and will basically contribute to the downstream body plan in larvae, for example. In contrast, here we have the same embryo, except one of its alleles is from the ancestral type, which is the sort of mismatch type. The other allele is, by the way, totally normal. Again, you'll go to see these synchronous rounds of division, but you reach a critical point in development at which you basically see that a lot of the nuclei start falling to the center of the embryo in a process that's called nuclear fallout. And that's happening because somewhere in their mitotic divisions, they actually made an error. But because these early embryonic divisions are happening very, very quickly, you do not basically block mitosis because there are no checkpoints active at this stage of development. Instead, you mark these nuclei by mechanisms which we still don't actually understand. And then at this critical point, juncture in development, these nuclei will lose their actin anchoring and fall to the middle of the embryo and no longer participate in development. The net result is that you reach the same stage of development with not the same number of nuclei required for development, and these embryos will die in another sort of 24 hours. So what I've shown you here is that we can not only identify evolutionary sort of processes that are driving unexpected rapid evolution of chromatin proteins such as SID, but we can use the same signature of rapid evolution to actually create these in vivo allelic replacements to understand what is actually driving this rapid evolution. In this example, I've shown you that when we replace the melanogaster SID with either the ancestral or the similan SID, we have lower viability and developmental delays, but also we have incredibly lower fertility when you do sibling crosses, which suggests that even though these females and males are capable of producing gametes, they cannot actually sustain early embryonic development in the absence of having the right SID protein. So a question that many of you are thinking about, which we are also thinking about, is what is the nature of this incompatibility between SID and the melanogaster genome? Is it in fact a DNA protein incompatibility? We believe that the answer is yes, because we are now doing cut and tag experiments thanks to Steve Henikoff's lab being next door, and we are actually indeed seeing that there is a different localization of these SID alleles. But we haven't actually ruled out the possibility that there's also an incorrect interaction with other proteins, including these chaperones that deposit SID, and this is something that we're actively investigating. I want to sort of remind you of what was initially depressing, but now sounds kind of very cool, which is in this cross where we have very little SID protein that comes in from the dad's pronucleus is actually sufficient to rescue embryonic development, even though the vast majority of SID protein in this zygote is coming from mom is of the wrong type. That's a remarkable example of an epigenetic rescue, if you will, where a little amount of the right SID protein is able to still educate the wrong SID protein to deposit in the right spot and basically propagate correctly. So after all of that, what I've shown you is that, in fact, it's not meiosis or centromere identity that is the deleterious consequence of rapid evolution of centromeric DNA. It's, in fact, embryonic mitosis in particular, but also later somatic mitosis with developmental timing defects that is actually basically braving the brunt. So this is just, again, an example of where we are now in evolutionary biology, where we not only have the ability to identify unexpected innovation in fundamental processes, but also try to understand using these allelic replacement experiments, what is in fact the driving consequence of these changes in vivo. I think I'm only got about 15 minutes, so I'll try to tell you as much as I can about a different example, and hopefully you can catch up with experiments. Many of you are also interested in this sort of contrast where chromatin is fundamentally different in most cells between somatic cells versus sperm cells. And that's because most DNA is packaged by canonical histones, which are very slowly evolving, but most DNA in sperm is packaged by protamines, which are very rapidly evolving. If you've never heard about protamines, they're this very cool DNA binding protein that occur in most animals, not all animals, in which you basically slowly replace the histones and their variants in the bulk of the genome with these very charged DNA binding proteins that are basically capable of further packaging the DNA up to 100-fold more tightly so that the genomic DNA can actually fit inside the sperm head. It's a remarkable example and is often critical for male fertility. I'll just show you an example of like how dramatic this difference can be. Here are two frog species. This frog species does not encode protamines. And you can see it has this very respectable bulbous looking sperm that is going to be capable of fertilization within the species, whereas this very closely related frog species encodes a protamine, and you can see it is now capable of this sort of very pointy streamlined shape, which is going to be obviously advantageous in terms of swimming speed and fertilization, and explains probably why protamines arose multiple times during animal evolution to give sperm a fertilization advantage over those sperm that don't encode protamines. So what I'd like to sort of also remind you is that protamines, especially protamine 1 and protamine 2 in humans, are in fact among the fastest evolving primate genes, in spite of the fact that these both of these proteins are absolutely essential for male fertility in mammals. And so what I'm showing you here is the sequence of protamine 1 from humans and chimpanzees. And each of these bold letters indicates amino acids where they are different. There are no synonymous changes because these species are so closely related. And so we have essentially a DNDS ratio that approaches infinity in this case. So we have a very good hypothesis to begin with about what is going on because there is sperm competition. There is, obviously an advantage to having a fertilization success. And so we can imagine that maybe the reason protamines are rapidly evolving is because they constantly confer this fertilization advantage, and that's the basis for the rapid evolution. But this doesn't make perfect sense because what we see is that even in cases where there are monogamous species, where there is no sperm competition from different males, we still have very rapid protamine evolution. And in fact, there is no correlation between the intensity of sperm competition and the intensity of protamine evolution. Moreover, there are many species, such as zebrafish, that have intense sperm competition but don't encode protamines at all. So this seems like an imperfect hypothesis. Moreover, when we take a closer look at protamine evolution in mammals, we realize that a lot of these codon changes are driven by changes at arginine residues, which are heavily encoded by codons that are rich in CpG codons, which, as you all know, are subject to hypermutation because of the fact that CpG codons are hypermutated because of methylation. So what we have here potentially is a very weird mutation selection balance where CpG codons are hypermutated and selection is now needing arginines to negate the negative charge of DNA, and that's what leads to this apparent rapid evolution of protamines. So we wanted to study this problem. This problem actually was very, very exciting 20 years ago, and then it sort of went sort of dormant for a while because this finding made it look like this might be an artifact of hypermutation at CpG codons rather than something interesting with protamines. So what we decided was we're going to look at this problem in Drosophila species because they also encode protamines, although their protamines are very distinct from mammalian protamines, but they lack DNA methylation. So we don't have to worry about CpG codon methylation as the driving signature here. But you can see that there's this beautiful ordered progression from histone and histone variant rich packaging to these beautiful needle-shaped sperm that end up in sperm, which are basically almost completely devoid of histones at this point. And this is occurring because they're slowly replaced by protamines that are basically carrying out this very important role of DNA packaging. So the first question we asked was like, are protamines in Drosophila also rapidly evolving, just like they are in mammals? And the answer is yes. So we can immediately say that the rapid evolution of protamines we see in mammals is not just an artifact of the CpG methylation. It's something biologically interesting but unexplained. Moreover, if you basically look at three of the protamines that are individually essential in Drosophila for male fertility, you can see that some of them are also among the most rapidly evolving proteins. So Drosophila melanogaster has 15 protamine genes, and MST77F, which stands for male sterile gene at the 77F cytogenetic location, is in fact the most rapidly evolving protamine gene and happens to be completely essential for male fertility in Drosophila melanogaster. So the question is why? And I'd love to take credit for any of this work, but in fact, all of this was actually pioneered by a former postdoc in my lab, Qingho Chang. So what Qingho did was the approach that I've already told you about, where we basically take this single locus and make an allelic series, hoping that the evolutionary sort of discordance between MST77F from different orthologs with the melanogaster genome might actually tell us what is actually driving this rapid evolution, including potentially sperm competition defects. What we realized was that MST77F, despite being completely essential for male fertility in Drosophila melanogaster, is itself a young gene. It only arose about 20 million years ago in these species of flies. So we essentially made an allelic series with each of these MST77F genes into Drosophila melanogaster. You'll notice that they actually very, very rapidly diverge in amino acids. So the Drosophila ananaceae gene, despite being only 20 million years separated, only has 22% amino acid identity because these are very, very rapidly evolving. So the first question is, are these capable of restoring male fertility? Because after all, that is the number one function that we associate with MST77F. And the answer is, for the most part, yes. So if we were to look at homozygotes encoding each of these replacement alleles from Melanogaster, Similans, Yakuba, Eugracilis, and Takahashii, this is roughly around 15 million years of evolution. They all can, to some extent, restore fertility. There is some modest drop in fertility in some cases, but the good news is that they're all fertile. Ananaceae, which is 20 million years separated, cannot rescue fertility. It's basically just as sterile as a knockout of MST77F in Drosophila melanogaster. So that's an indication that MST77F rapid evolution has now gone past the threshold where it can rescue fertility. This is a little misleading, however, because if we were to look at heterozygous males with only one copy of MST77, you'll notice that melanogaster is the only allele that is able to restore fertility in a heterozygous state, whereas the others are all incapable or haploinsufficient for male fertility. What we basically then asked was like, what is actually going on with the transmission of all of the chromosomes? In particular, what is going on between the transmission of the X and the Y chromosomes in these males that encode these replacement alleles? Because you'll recall when we cross these, we should get males and females in roughly equal proportions if both the X and the Y chromosomes are basically inherited under Mendelian proportions. But instead, what we see is that is only true when we have the melanogaster homozygous males, where we have close to 50% progeny, whereas in the other replacement alleles, we get a lot more males than we do females. What that means is, I just want to sort of think about this, is that just like in the SID experiment, these flies are completely identical, except as a single locus, where we've basically taken swaps between this rapidly evolving protamine and swap them. And what we've unleashed is a dormant sex ratio driver that basically accelerates the rate of male production over female production, because you're presumably having many more successful Y-bearing sperm that are transmitted than X-bearing sperm. And this is actually happening because we have a fundamental difference in DNA packaging, which we basically almost stumbled upon by accident. Because in a typical Drosophila sperm, we can barely get any antibody staining. This is actually the bane of my existence, because cytology becomes very difficult in sperm because they're so tightly packaged that antibodies basically are sterically hindered. But actually, what we discovered was even antibodies to double-stranded DNA begin to light up in either hemizygous males with melanogaster alleles or in the homozygous males with the wrong sort of species alleles of MST77F. And then what we took advantage of is the fact that in Drosophila, if you pay attention to this here, all the X and Y bearing sperm are produced at the same time in a clutch from the same cyst, because all products of the same meiosis produce sperm in the cyst, which are basically tightly sort of linked to each other. What that means is that we can ask in the same cyst that contains both X and Y chromosomes, do we see evidence of decondensation, which we do by staining with double-stranded DNA, and we can use FISH probes to the X or the Y to ask, is it more likely to be the X chromosome or the Y chromosome that's decondensed? And what we find is that there's an overwhelming amount of decondensation happening with the wrong MST77 allele on the X chromosome. What that means is that when we look at mature sperm from each of these males, they're basically massively enriched in Y-bearing sperm and have depleted X-bearing sperm, because those have basically been decondensed and not finished all of the development needed to become mature sperm. As a result, what we basically now have is a unique hypothesis where protamine evolution is not driven by a competition between sperm during fertilization, but in fact is driven by competition between sex chromosomes during spermatogenesis, where autosomal protamine genes are suppressing this behavior. Chingo was very inspired by this sort of observation and asked, what about other Drosophila species? By the way, I should say again, for full disclosure, I absolutely forbade him to do this experiment, but he wanted to know whether Drosophila melanogaster was unusual in showing this sex ratio bias. So he decided to look at Drosophila jacoba, which is only 6 million years separated. We don't have exactly the same tools in Drosophila jacoba, but we can create knockouts, and that's what he did. And I thought that these males are going to be sterile. But to our surprise, these males are not sterile. So actually, the one fundamental property that we know to be true for MST77F is it's completely essential for male fertility in Drosophila melanogaster. But in a very closely related species, it's not essential. It's important because you see this drop in fertility, but it's not essential for male fertility, which means even the male fertility nature of protamines varies dramatically even between closely related species. But you can ask, in this homozygous, heterozygous, or knockout male, what is the sex ratio bias here? And what we can see is in the homozygous male, we have sex ratio bias consistent with Mendelian segregation, and it gets more and more skewed the less and less MST77F you have in the spermatogenesis. So what that means is that it's actually not male fertility, but suppression of sex ratio, which is actually the primary driver of MST77 function that is occurring in Drosophila species. So we started off with this genome packaging function and sperm competition as the hypothesis. But what's really nice is that we have this allelic replacement approach, which basically tells us that it's actually this genome silencing function of silencing some sort of sex ratio distorter that is actually the main purpose. And these competitions between distorters and suppressors are driving this competition. So we start with this focal observation that autosomal protamines, or SNBP genes, are suppressing competition between X and Y chromosomes. Actually, we also uncovered about 19 cases, which I don't have time to tell you about, but there's a nice paper in eLife that Chingo has published, where these protamines can land onto the sex chromosomes and expand in an accordion-like state to become dominant negatives that become drivers themselves. So you go from being a peacemaker to becoming a rogue by becoming linked to a sex chromosome. and one very remarkable species in which the X and the Y chromosomes have fused to each other. And when that happens, anything that kills that the X encodes that kills the Y or vice versa becomes actually a suicidal gene. And so those suicidal genes are lost because of the XY fusion. And what we see is that even genes that are essential for male fertility, like MST77F, have been spontaneously lost, suggesting once again it's not their sperm condensation function that's keeping them in Drosophila genomes, but it's actually their need to suppress and maintain equal sex ratios that is actually keeping them in the population. So what I've shown you here today are two vignettes of examples of chromatin genes in which we basically have found signatures of rapid evolution, and that prompted an exercise to try to identify what is driving the rapid evolution. And in both cases, we uncovered genetic conflicts occurring during or just subsequent to meiosis that is driving that. We have actually a couple of other protein categories that we've published on or are actively working on. I'd be very happy to talk to you about that. But I want to return to this sort of example where, again, we have this dichotomy of the cell, and it's actually telling us that the explanation for why genes that are essential for cell biology and yet evolve rapidly is actually that there are intrinsic genome-borne selfish genetic elements that are constantly perturbing the equilibrium and forcing these important proteins to adapt. I think I'll stop there, and I'll be very happy to take questions. Thank you.
Oh, thank you, Harmit. If there are any questions, feel free to type them into the chat or go ahead and unmute yourself and ask questions.
Hi,
Don't feel it. Yes, don't feel intimidated,
No.
even though this is new.
Hi, Harmit. Thank you so much. This was absolutely great talk. Thank you very much. I do work in corals, and so it's a little bit different platform, and certainly it's more epigenetics than anything else. But I love understanding, better what genetics can allow you to do, and especially the mechanistic analysis that you bring in. I had like a couple questions. I don't understand very well how is that this mismatches or this the centromeric DNA and cytochore proteins contribute to hybrid inviability. So how can you be looking at the centromere histone, and how would that lead to hybrid inviability? Or
Actually, I'll just answer that question by showing you these slides, because literally my next slide was that this can lead to inviability, because essentially what we have is centromeric DNA and proteins that can rapidly diverge, not just at centromeres, but also at other satellites. And in fact, that could be very well the process that leads to postzygotic isolation, that leads to sterility and inviability defects. And even though a lot of this work has been done in Drosophila, so there's a very strong Drosophila bias in speciation research, 7 out of the 8 mapped animal hybrid incompatibility genes, which means genes which, when mutated, will actually recover the fertility or viability of the hybrids, themselves turn out to be either satellite DNA or satellite DNA binding proteins. So it's very, very clear that the sort of impetus for postzygotic isolation, or this mysterious phenomenon where hybrids produce either infertile or inviable offspring, is actually probably emerging from these genetic conflicts that are occurring in these satellite-rich parts of genomes.
That is so interesting.
Thank you for giving me the opportunity for the 2 slides I didn't have time to present.
My other question was, it's all big picture, because this is fascinating to me. I was not even aware of these nuances. So if centromeres all perform the same essential function, how do you reconcile the fact that the associated proteins are evolving so rapidly? They're all doing the same thing. And it's very important. I see your point between keeping it conservative, because you're producing the same thing over and over again is so essential with this rapid evolution.
I think the key in the case of the centromeric proteins is that the equilibrium is constantly. So for example, if you were to look at budding yeast, budding yeast have only symmetric meioses, and their centromeres are very simple, and the centromeric proteins are pretty much equivalent, if you will. But budding yeast don't have female meiosis, but it's actually female meiosis in plants and animals that gives centromeric DNA the advantage of playing this game where they can increase their transmission over their other sort of homologs. And because of that, you constantly perturb the equilibrium, and then the centromeric proteins are trying to restore the equilibrium. So essentially, the Red Queen conflicts are all about one party taking advantage selfishly of the opportunity that is provided, and then that is often in cis, and then the transacting party is often acting as a suppressor, and that's kind of what we see with the protamines as well, where there is a constant battle between the X and the Y bearing sperm because they are genetically distinct from each other, and they can actually drive one, to the disadvantage of the other. But now the transacting protamines are actually trying to suppress that competition because for the species, equal sex ratios is important. But for that particular X chromosome, it's actually advantageous to like outcompete the Y chromosome because then more of it will make it in the next generation of X chromosomes. So what we have is like essentially selfish interests that perturb the equilibrium, and then global interest, if you will, that try to restore the equilibrium. And that's probably what's going on repeatedly. Probably the same thing is going on, for example, with polycomb proteins and suppression of transposable elements, where with transposable elements, it's quite easy to understand that they can actually act selfishly by increasing in copy number at the expense of genome stability. And therefore, all the proteins that suppress transposable elements are now acting as the sort of the transacting suppressors, whether the transposons themselves are cis-acting selfish elements.
I see. That's very good. Very good. Very helpful. Thank you so much. Very, very helpful.
Thanks.
Thanks, Luisa. Any additional questions?
You're also welcome to email me because I know time is short and we are already at the hour. So please feel free to email me. Both of the things that I presented are actively being written up or revised as well. So we'd love to get your feedback.
All right. Well, we'll keep it close to time. And thank you again.
Thanks for the opportunity. Thanks for listening. Bye-bye.
And bye-bye. Take care.